Hello everyone, welcome to another video of class 10 General Science, and today we are going to continue with heredity and evolution, and today's video is going to be on the dihybrid cross of Mendel. So without wasting any more time, let's get started. In our previous video, we had talked about the monohybrid cross. So this time we are going to talk about the dihybrid cross. And why is it called the dihybrid? Because we are taking into consideration two characters. In our previous video, if you have not seen that video, then I would like you to go and check out that video in my channel, a very important one in order to understand this video. So we are taking into consideration two characters in contrast to only one character which we took in the last video. We took the character of height. This time we are going to take the most common that Mendel took to understand the dihybrid relations, and these characters are the seed shape and the seed color. These are the two characters that Mendel took while considering how two characters will be expressed together. Okay, so here seed shape can be of two types. It can be either round or wrinkled. And seed color can be of two types. It can be either yellow or green. Now, as we talked about the dominant and recessive characters, in the seed shape, the round is the dominant one and wrinkled is the recessive character. Yellow is the dominant in case of seed color and green is the recessive one. Okay, that means if round is present in presence of wrinkle, then round will be expressed and not wrinkled. Similarly, if yellow is present in the presence of green, then yellow will be expressed and green will not be expressed. So for round we use capital R, and for wrinkled we use small r. Okay, dominant is capital, recessive is the small letter. In case of yellow we use capital Y, and in case of green we use small y. So these are all about the samples. Now let us see how the cross was done. Now it must be in your mind that in the book the whole process is not given, but I would like to explain you the process so that you understand what actually the thing is happening. So Mendel chose in the same way as monohybrid cross, he chose two parents which are of homozygous character, homozygous dominant and homozygous recessive. Means the first plant that he chose was round and yellow. The seeds were round and yellow in color. So round means capital R, both dominant, and yellow means capital Y. So two pure characters together. If it had been monohybrid, we would have taken only capital R, or suppose only capital Y. Since it is dihybrid, we will be considering two characters at the same time. So pure means homozygous dominant round and homozygous dominant yellow. This is one of the parents, and the other parent will be homozygous recessive, means all the alleles or all the characters will be in the recessive form. So it will be wrinkled, small r, and green. Okay, so that is, these are the two parents and we will cross them. So this is the P generation. Okay. Now after crossing these, what are the gametes that will be formed? First we need to look at the gametes, then we can see the offsprings. So what are the gametes? Here we will have one allele from each of the characters, right? As we formed in the last one, from capital T, two gametes are formed, capital T and capital T. Right? These are two separate gametes. Here also from R we are going to get one R, and from Y we are going to get another. So it is R and Y. Okay? Since it is two characters, so we are going to take one from each. Okay, I hope that is clear. So what are the gametes that we can get? We can get R, Y, this and this, or we can get this R, this Y, or we can get this R and that Y. So everything will be capital R, capital Y, capital R, capital Y. Everything will be capital R, capital Y. From here again, everything is going to be one small, both small r and small y, right? One from, suppose this one and this one come together to form this. Or we can get this one and this one coming together to form this, or this and the first and the fourth. So all are going to be same. Second and third again RY. So all are going to be the same type. So rather than writing 4 in a row, let us just write what we are going to get. So this is small r, small y. When these are combined, the gametes that we actually get are capital R, small r, one capital R from here, one small r from here, one capital Y from here because there is no other option, and one small y from here. So this is our first generation, F1 generation after the combination of the gametes. So what are the gametes? The gametes are capital R small y, capital Y and R Y. Okay, so rather than giving it like this, let me put this. So combination of these gametes will give capital R small r, capital Y small y. So what is going to be the character of this one? Capital R small r will give us what? One dominant, one recessive, means the dominant one, that is round, and this one is going to give us yellow and green together, means obviously yellow because yellow is dominant. So this was round and yellow, this was wrinkled and green. What do we get? We get round and yellow, means the dominant characters are expressed just like in case of monohybrid cross. So again the same situation, we took two homozygous, one dominant and one recessive plant, and what we got was a dominant character, no recessive character is being shown. Now we are going to go for the most important part, that is the second generation. How to obtain the second generation, that is the part that is different from, or a complex part compared to the monohybrid. So let's see how to do. For doing this, we will use one square, just like a matrix box. We will use a square which is called the Punnett Square, P-U-N-N-E-T, Punnett Square. So Punnett Square means it was invented by Punnett, the scientist named Punnett. So Punnett Square we will use. So for this we will need to know what are the gametes that we can obtain from this one. This is the offspring that is formed. So from here what we can obtain? Either it will be capital R from this one and capital Y from this one. This is one possibility. What is the second gamete? Capital R and small y can be one of the gametes, right? What is the other one? Small r and capital Y can be one. And what is the small r and small y? So we have 4 gametes here. Okay, so these 4 gametes can combine in different ways. So we are going to have 2 plants, obviously for, the same plant we are going to do a self-fertilization. Means this plant we are going to fertilize it by this one, just like monohybrid cross. So that's why I'm asking you, please go and watch the monohybrid cross video, then you can understand, because I'm going to take reference from there. Okay, so we have these gametes, possible gametes. So we are going to have 2 sets, right? Because one set cannot fertilize. So we are going to have 2 sets of these, and they will have a cross among themselves. So let's see how to do this. For this we will use the Punnett Square. So I am erasing this whole part and we are going to use these things. Okay. So what we will draw is a 4x4 square, 4 columns and 4 rows. Okay, 4 columns and 4 rows, 16 box square. Here we will write the gametes for each box. Okay, so suppose this part is for the female part and this part is for the male part. So whatever we are going to write here is going to be for the male part and this is for the female plant, obviously. So what are the possibilities that we had? Capital R, capital Y, capital R, small y, small r, capital Y, or small r, small y. Okay, here what we are going to have, okay, let me get this. Yeah, here again we are going to the same possibilities, right? Same set. Okay, so now we will find the cross, because in this box what we will write? We will write the combination of these two. This and this combined together to form capital R, capital Y, capital Y, capital Y. Easy? Let's continue. Capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Again, capital R, this and this will give us the second box here. Capital R, capital Y, small y, capital R, small y, small y, capital R, small r, capital Y, small y, capital R, small r, small y, small y. Again, capital R, small r, y, y, again r, r, y, y, r, r, y, y, and here we are going to get r, r, y, y. Last one, capital R, small r, capital Y, small y, capital R, small y, small y, small y, small r, capital Y, small y, and small r, small y, small y. So these are the 16 possible offsprings that can be obtained. Now let us see what are the characters. What are the characters? For this one it will be round and yellow. I am writing short form, okay, round and yellow. This is going to be again round and yellow. This is also round and yellow, round yellow. This one again round yellow. This one will be round plus 2 small y means green, recessive. This one will be again round yellow. This one will be again round and green, 2 small. Again round yellow, round yellow. Smaller means wrinkled and yellow. Again, wrinkled and yellow. Here round and yellow. Here round and green. This is wrinkled and yellow, and this is wrinkled green. So these are all the phenotypes, means what we can see as the outcome. Now, how many of each type? Round and yellow means both dominant. If you count 1, 2, 3, 4, 5, 6, 7, 8 and 9. So 9 offsprings are round yellow. Round and green, we are going to get 3. See, 1, 2 and 3. If you go for wrinkled green, wrinkled and yellow. Okay, wrinkled and yellow means WY. 1, 2, 3. And finally, wrinkled green, only 1. So total how many? 9 plus 3 plus 3 plus 1 is 16. And here we have total 16. So out of the 16, the phenotypic ratio will be 9:3:3:1. Round yellow 9, round green 3, wrinkled yellow 3 and wrinkled green 1. So what is the phenotypic ratio? Phenotypic ratio is 9:3:3:1. This is the phenotypic ratio that we obtain from the dihybrid cross. Okay, now if we go to look for the genotypic ratio, a very complex one, but what you can do is, I am just writing the ratio because it will be time taking to count. So for each type, suppose capital R, capital R, capital Y, there is only 1 here. You will not find same anywhere else. So it will be 1:2. Then if you take this one, capital R, capital Y, small y, there will be 2. So if you go on doing like this, you will get this ratio. This ratio you are going to get. So what is the ratio? 1:2:1:2 4 2:1:2:1. Very easy to remember. 1:2:1:2. Then you write 4. Then again it 2:1:2:1. That is the genotypic ratio. It will not be that important for class 10, but this ratio will be important. This is the ratio of the phenotypic characters. Okay. So that is all about our dihybrid cross. This is our F2 generation. Here we get the phenotypic and genotypic cross. So what is the difference between the monohybrid and dihybrid is that we took only one character in monohybrid. Here we took a combination of 2 characters. Okay, and we found out the outcome. So what is the use? We got the third law of genetics. That is Mendel's third law, which is the law of independent assortment. Law of independent assortment means even if there are multiple characters, as we took 2 characters here, even if there are multiple characters, the presence of one character will not affect the presence of the other character. Suppose it is not like if there is a round gene, then the color will always be yellow or the color will always be green. There is no dependency. If the seed is round, the color of the seed can be yellow or green, anything. So there is no dependency that if there is capital R, there has to be a capital Y. No, here we have capital R. Suppose this one, we have a capital R, but we have 2 small y's. So it can be round and green, it can be round and yellow also. It can be green and wrinkled, it can be yellow and wrinkled. So everything is possible. It means that the presence of one character does not affect the presence of the other. So how one character is assorting itself, arranging itself, does not affect the other character. So they are not dependent on each other. That's why it is called the independent assortment. Every character will arrange themselves independently without the effect of any other character. Okay, so this is what you learn now. I'm just giving you hint because there are exceptions in everything in biology. So what you are going to, if you take biology in higher secondary, you are going to see that this is not always true. Sometimes the presence of one character affects the presence of the other. Okay, so suppose you have one character in your body. I am just taking a random example. This is not correct. I mean, just for example I am taking. Don't take it seriously. This is just for an example. So suppose a character of eye color and hair color. So it may be something like you will get that if the color of the hair is black, your color of the eye will never be black. Suppose, just an example. So that means the color of the hair will affect the color of the eye. So in like this, you will find some characters where presence of one character will directly affect the presence of the other. So these things we are going to learn, but as of now, the law of independent assortment means presence of one character will never affect the presence of the other. So that is all about the dihybrid cross. I would like to add one more part because it is a very small topic. So let me add the sex determination in human beings. That is a very easy topic and I am trying to insert it in this video. Sex determination in human beings, very easy. So sex determination, whether a human being is going to be a male or a female, that is determined by the X and Y chromosomes. Very simple. You must know that this is there are 23 pairs of chromosomes, right? I told you in the first video there are 23 pairs of chromosomes, out of which 22 pairs are non-reproductive. They don't take part in reproduction, but one pair takes part in reproduction, and this pair will determine whether the organism is male or female. So as I told you, the chromosomes always stay in pairs, right? 22 pair, one pair. So in this pair, the chromosome stays in pairs. So chromosome shape is like this in the form of X. So if you have two X chromosomes, then the person is going to be a female. This is the symbol of female. Okay. If this chromosome is of the form of XY, then it is a male. So if it is XY, it is a male. So that is how we determine whether an organism is going to be a male or female. So XX means male and XY means female. Why XY? Y means its original form was XX, but due to some mutation, that is change in the chromosome, this one arm of the Y got deleted. It was removed from the cell, from the chromosomal part. So as this part was removed, it looks like Y. That's why the XY means a male organism, XX means a female organism. Now what is going to be the sex of a child? Suppose a father and a mother, they are trying to reproduce. So the father will be XY and the mother will be XX, and we are trying to do a cross between them. So what are the possible gametes? Gametes will be either X or Y. Here gametes will be both X, right? So now we want to combine them. What are the possibilities? Either this X with this X, XX, or these two, or these two, and these two. So these two are going to be female and these two are going to be male. So the ratio is always 1:1. So XX means male, female, and XY means male. So that is how, this is our parents, this is our gametes, and this is our F1 generation. So what do we understand from this? What we understand is whether an organism is going to be male or female, it depends on the father, not the mother. Because a mother will always give an X chromosome. Mother can never give an Y chromosome. And what determines an organism to be male or female is the Y chromosome. If the Y chromosome is present, it will be a male. If the Y chromosome is absent, it will be a female. So the father is the only one who can give the Y chromosome, and father only causes a male organism. If the father gives X, it is going to be female. If the father gives Y, it is going to be a male. So it is always the father who determines whether the organism is going to be male or female, not the mother. So there is a misconception. I am telling this because in many parts of our country we see that if a female organism is, a female child is born, then we always where the birth of a female child is protested. And there are many parts of the country where we see this, and they always blame the mother that the child is male or female. That is not true. The mother is not the cause of a male or a female child. It is always the father. So first of all, it is just a pathetic thing to actually demean either male or female child, because we need both male and female in our society. It is not a male dominant society. It should not be a male dominant society. In many villages you will see female children are born and they are not accepted by the society, and they always blame the mother. So first of all, I would like, as I am trying to teach this subject, I think we must remember the social aspect of our life, and this should not be the case in any part of the country. We should not support this thing in any part of the country. We should support both male and female child, and in addition to that, never blame the mother for anything. I am not asking you to blame the father, but never blame the mother, because scientifically it is proven that the mother has no part to decide whether an organism is going to be male or female. It is always the father. So keeping this social side in mind, and hope you understood this video, a very interesting topic. So if you have understood, then make sure you hit the like button, and in addition to that, if you have any confusion, then make sure you ask me in the comment section, or you can give me a call or wherever you want. So thank you everyone for watching, and until next time.